Hi, hello. My name is Adolfo Garcia-Sastre, and I'm professor at the Department of Microbiology and director of the Global Health and Emerging Pathogens Institute at the Icahn School of Medicine at Mount Sinai. And today I will be presenting some of the data that we have from our group on development of new vaccines and antiviral treatments against COVID-19. All right, so let's start first with therapies. For COVID-19, there are not so many therapies that have been available, and there is still a lot of problems trying to give something to people that will reduce the burden of the disease, reduce hospitalization, reduce possibility of having severe disease, etc. There are some therapies that have been approved. One of them is remdesivir. This is an inhibitor of the viral RNA-dependent RNA polymerase, but is not working 100% in terms of preventing severe disease. There are some others that have been approved for emergency use and that have been used in the clinic, such as the plasma from convalescent individuals or the existing monoclonal antibodies, although there is some concerns that the new variants may not be able to be neutralized by some of them. And these, the ones that I've mentioned till now, they are treatments that reduce virus replication. These type of treatments, they are more important to be given early during infection because you want to stop the virus from replicating early on. Once that people develop severe symptoms, there is less remaining virus replication. Most of the disease now is being caused by an exacerbated pathological response in the host, and therefore inhibitors of virus replication, they don't work so well. What we need at this time is inhibitors of the disease itself, of the symptoms of the disease. And among the ones that have been used is the one that is more popular is dexamethasone, which is an immunosuppressive drug that seems to at least reduce some percentage of mortality as well as of time that the people are required to be in the hospital with ventilation when being used in the hospital for people that start to have severe symptoms. But there is a lot still that is needed in this area. We don't have a really very good working treatment, and therefore there is still a lot of studies that are being conducted trying to find new treatments against COVID-19. Now, the type of studies that we have been involved on, and not only my lab, but many other labs, they have to do with what is called drug repurposing. And drug repurposing is based on the fact that there is a lot of already existing drugs that have been used in humans or that have been used in clinical trials in humans, for which we know already some of the properties of these drugs in terms of for how long they stay in the body, what concentrations have been achieved in the body when they have been given to people, what type of adverse events they are giving to people. And these are some information that we already have, and that if among the drugs that are already being used in humans against some ailments that they don't need to be even viral infection, or that they have been tried in clinical trials, and then we know already some of the properties of these drugs. Among these drugs, there is one or several that allow also for inhibition of viral replication. These drugs can be deployed more quickly because we know already some of the properties when they are administered into people. And why a drug that is being used for a different ailment will make an impact in viral replication? Well, viruses, they require to use host factors to replicate because they are parasites and they require these host factors. And they require a lot of host factors. There are a lot of things that the virus miss, and that's why they need to come into a cell and take many of the host factors that they require in order to make copies of themselves. Now, if among the host factors that the virus requires, there is already a factor for which a particular drug is being used already in humans for some other treatment, some other ailment, and just happen to be that this drug inhibits the same host factor that the virus requires for replication, then this drug will make an impact in virus replication by inhibition of this host factor. So that's the idea about drug repurposing. Now, there are several ways how you can try to find drugs among drugs that have been already been used in humans or they are being used in humans. One of them is to screen all of them. Our lab has decided to go through a little bit more of a focused assay in collaboration with many other people, which is making some predictions about what of the drugs that are around they are most likely to make an impact in viral replication. So, and I will tell a little bit later in 1 second how we make these predictions. But once that we have these predictions, then we take these drugs and then we test it for the ability of these drugs or not to inhibit the virus in tissue culture. And if that allow us to find now some drugs that inhibit the virus in tissue culture, and from that, the ones that are working best, we try to use them in animal models to see whether they are efficacious in an animal model, and move hopefully quickly into clinical studies in order to see that at the end whether they make an impact on the disease. So how we make these predictions? So that can we identify compounds that inhibit host factors involved in SARS-CoV-2 replication among known human drugs? And for that, we have been collaborating with UCSF, especially with the group of Nevan Krogan, which is a person, it has a group that is interested in protein-protein interactions and protein modifications and how these shape the different diseases, including viral diseases. And what he did very early on, as soon as the sequence of the SARS-CoV-2 was posted online, and knowing that this was the causative agent of COVID-19, he cloned each individual viral protein encoded by SARS-CoV-2, a total of 29 proteins, and then expressed them in human cells separately, and then determined through immunoprecipitation and mass spectrometry the host proteins that are interacted with each of these viral proteins. This is what is called a virus host interactome map that we can see here in the middle of the slide. And what you can see is little dots, red dots, that are having a constellation of all the factors. It's too small to be able to read here, but it's sufficient to say that each small red dot is one viral protein, and the constellation around are the host proteins that are interacting with these viral proteins. And the idea is that if a viral protein is interacting with the host protein, it's likely that it's doing that because it wants to use this host protein to be able to allow him to replicate. So we think that among these 332 protein-protein interactions map, there is an enrichment for host proteins that are required for viral replication. Now, is there any of these 332 proteins, is there any that we know that can be inhibited by one known human drug that is being used for another type of treatment? That's where we came out with a total of 69 factors for which there are known drugs that can be inhibit these host factors that we found interacting with viral proteins. Now, for now, identifying drugs, we collaborate with Kevin and Brian, also at UCSF. They are chemical bioinformatics, that they have mined the spectrum of drugs that have been used in humans, for which ones they are able to inhibit any of these factors that interact with one of the proteins of SARS-CoV-2. And with that, they identified 97 different drugs that we tested for inhibition of viral replication. And in my lab has been, in my group, Chris White, together with Romel, Elena, and Lisa, the ones that have been looking to these compounds that Kevin and Brian found that are some evidence that they will inhibit one of these host proteins that interact with one of the viral proteins. Now, the assay that we use is a medium throughput assay in which we use live viruses, SARS-CoV-2. We use a replication assay in biocontainment. We seed first cells that will support replication. In the beginning, we use the ones that we know early on that supports well replication, which are Virology cells, which they come from African green monkey. And then these cells have been seeded into little wells. Now we put in these wells the different drugs that we're going to be tested at different concentrations in triplicate, and then we infect them with SARS-CoV-2 with an amount of viruses that is around 5 viruses per 100 cells. So you get an infection of only a percentage of the cells, and then the virus starts to propagate, and in 48 hours, in the absence of any drug, the virus is infecting almost any cell of the culture. Now, if there is a drug that inhibits viral replication, then it's going to reduce the number of infected cells, and that's what we measure at the end. At the end of 48 hours, we fix the cells, we inactivate the virus, we take these wells from the biocontainment, and then we measure the extent of viral replication in our group by looking at the number of infected cells using staining the infected cells with an antibody against the virus nucleoprotein. And if we see that there is an impact, then we also determine the virus that have been produced in the supernatant to see that actually there is an inhibition of viral replication, not only the number of virus-infected cells. And we have done these assays together or in parallel to with a different group that is in the Institute Pasteur, Olivier, Marco, and Christophe, that they have used a very similar assay, but they use as measure viral replication, viral RNA loads. And we have very concordant results, which actually give us confidence that what we are seeing is actually true inhibitors of viral replication. All right, so here I'm going to show you an example of how remdesivir looks like in this assay. So we use remdesivir at different concentrations, and then we look for the ability of these different concentrations to reduce the percentage of infected cells. In the absence of remdesivir, we have 100% of infected cells. As we increase the concentration of remdesivir in these wells, we are starting to see a drop in the percentage of infected cells, which is depicted here by the solid line. And you can see that around 1 micromolar concentration, you get almost a complete inhibition of viral replication of virus-infected cells. And in the line that is not solid, we perform at the same time, in parallel, in non-infected cells, cytotoxicity studies. And what you can see is that remdesivir is not cytotoxic, the cells are still alive, but it's a good inhibitor of viral replication with IC90 inhibitory concentration 90%, around 1 micromolar, which is what you want to start with an inhibitor in order to see whether they can impact these concentrations more or less better, even if it's lower concentration. Now, these in viral cells, in human cells, 293 T cells that are human, that they are not infectable because they don't have the receptor T cells, but you can make them infectable if you express the human AC2 receptor for the virus. Now, in human cells, remdesivir is even better. It has an IC90 of 24 nanomolar, which is a very good one, and that's one of the reasons why this is a good inhibitor and why it's being used now in the clinic. It's a good inhibitor of viral replication. Now, we have tested all of these compounds. This is just an example of some of the compounds that we have tested and all the things that we have. In total now, with different collaborators, not only the ones that we have done with these studies in Interactome, but also some of the collaborators, we have tested more than 800 drugs. And from them, approximately 10% they are able to inhibit viral replication, and a few of them, they have a very good ability to inhibit viral replication. Now, amongst the most potent inhibitors that we found this way of viral replication are compounds that are known to inhibit translation protein synthesis. This is one of them, Subtyphine, is able to inhibit one factor, host factor, involved in protein translation, EYF4A. And this factor is known to interact with one of the viral proteins, so that's one of the reasons why we use Subtyphine. It has been used in clinical trials for phase 1 and 2 clinical trials for the treatment of cancer, because cancer, if you inhibit protein synthesis, the cancer cells will stop dividing. And it has an IC90 in terms of inhibition of SARS-CoV-2 replication of 154 nanomolar. So it's a quite good, quite potent compound, and this is one of the ones that we probably will continue using it in clinical trials that will start soon. Now, other translational inhibitors that we have tried, they target a different factor, which is this EF1A. It's also involved in translation, and is also known to interact with at least one of the viral proteins. And among them, we tested Ternatine 4, which has a very good IC90. Now we are talking about 10 nanomolar, very low concentration required for making an impact on viral replication. And plitidepsin, also known as aplidin, is another compound that also targets EY1A and has been approved in Australia for the treatment of multiple myeloma. This one has an IC90 even better, of 1 nanomolar. It's very, very potent inhibitor, which is one of the things that you want to have when you're looking in a drug in these assays. Very potent inhibitor for a drug that has been already been used in humans, and we know that adverse events with respect to treatment, at least, of multiple myeloma. We see, if you look to the cytotoxic data, the ones in the discontinued line, it looks like these compounds, they're a little bit cytotoxic because you get reduction to 50% of the cells, but it's not that they are killing 50% of the cells that have been treated. It's because during the time of the assay, the cells double, if in the absence of inhibitor, but in the presence of this inhibitor, the cells don't double, they stay the same. They don't die, but they don't double, and then at the end of the experiment, if you compare them with not treated cells, there is only 50% of cells. But it's not because 50% have been killed, it's because this 50% has not doubled to 100%. So that means that perhaps we can also not have cytotoxic effects when we use them in vivo from the cells or adverse events if they are used at the right concentrations for still inhibiting viral replication. Okay, so these in vivo cells, now we move also to human cells to name 3Ts. And here we're comparing now remdesivir with plitidepsin or aplidin. And what one can see is that the IC90 of plitidepsin or aplidin in 293T cells, human cells, is sub-nanomolar, which is around 20 to 30 times more potent than the IC90 of remdesivir in the same cells for inhibiting the viral replication. So it's more potent than remdesivir. Not only works also in human cells like 293Ts, which are tissue culture cells, but it also works in primary cell human cells. These are here in the right corner on the top. These are pneumocytes, human pneumocytes, the ones where the virus replicates, that are derived from pluripotent, human pluripotent stem cells. And in this assay, remdesivir also is able to inhibit very well at the low concentration, 10 nanomolar, the infection by SARS-CoV-2. And at the end, we did what is called a time to addition assay, which is what is seen here in the right bottom corner, in which we treat cells before infection or after infection, and we see whether the inhibitor still is giving an impact on virus infection. And aplidin, even 4 hours post-infection, is able to reduce viral replication at the time that remdesivir is unable to, you need to treat early with remdesivir to make an impact. So it looks like in these assays, aplidin or plitidepsin is more potent than remdesivir. But the important thing is what the protein does in vivo. Now, plitidepsin is targeting EY1A, but there is always possibility that it's off-target activities of inhibitors, perhaps inhibiting viral replication, not by inhibiting EY1A, but by doing something that we don't know that plitidepsin is doing. Well, it turns to be that because plitidepsin and these inhibitors of EY1A, they have been used for the treatment of cancer, some cancers have developed resistance to this drug. And this resistance is due to a mutation in EY1A that changes one single amino acid, A399 by B, and prevents binding of plitidepsin to EY1A and then inhibition. Now, what we have done is we have made a cell line in which we have CRISPR-edited EY1A to introduce this mutation, A399V. And then we look whether plitidepsin is still able or not to inhibit viral replication. That's what we see in the upper right corner. In the upper right corner, we see that in red, cells that contains this point mutation, single point mutation EY1A, now they are unable to be inhibited by plitidepsin with respect to replication of SARS-CoV-2. Well, if we use remdesivir in the same cell, remdesivir has no problem because it has a different target. So what these experiments demonstrate is that the target for inhibition of viral replication of plitidepsin is indeed EEF1L. Remdesivir is being used, so probably for new inhibitors, they will be using in combination with remdesivir. So it's always good to see whether new inhibitors they are synergizing or they have additive effects, or perhaps antagonizing, so they don't work, they kill each other with remdesivir. In the case of aplidine or plitidepsin, we did experiments in which we combined remdesivir with plitidepsin or aplidine, which they are here summarized. And in this case, what we have is an additive effect. That means that one drug, the two drugs can be used, and not only that, because it's an additive effect, probably you need a little bit less of each drug in order to make the same impact. Now, does it work in vivo? For that, we have done some mouse studies. Mice are not being infected, at least with the SARS-CoV-2 that they were being circulating before variants came, and that's the experiment that we have done here is before the variants. And for that, we need to make, we can make mice able to replicate the virus, but we need to do tricks. And the tricks is make them to express the human AC2, because the mouse AC2 is not very good to allow replication of SARS-CoV-2 in the mouse. You can achieve that by transducing mice in the lung with a adenovirus vector that expresses human AC2, and 5 days later, after this transduction, now the lung cells of the mouse are expressing human AC2, so you can challenge them with SARS-CoV-2. When you do that, now SARS-CoV-2 will replicate, and replicates, as we can see here in the right, to levels around between 10 to the 5, 10 to the 3 PFU per ml at day 3 post-infection. Now we have taken these mice and treated with aplidine or plitidepsin or remdesivir, different dose, 1 mg per kg, one single dose, 0.3 mg per kg, 3 doses, and compared with remdesivir, 50 mg per kg, 3 doses. And what we see is that both remdesivir, but importantly plitidepsin, at as low as a smaller concentration, is able to reduce viral replication to a level that in some instances we cannot even detect viruses in the lungs of these animals. The other system that we have used is not a transduced mice, but a transgenic mice. In this case, these are transgenic mice that they are available from Jackson laboratories that express human AC2 under the control of a keratinocyte promoter, K18. And in this case now, because this results in better expression in a lot of cells of human AC2, the replication of SARS-CoV-2 in these animals is much, much higher. It goes to 10 to the 7 PFU per ml. And when we did the same treatment here at 0.3 mg per kg 3 times, we again were able to reduce dramatically the replication of the virus, indicating that, yes, aplidine or plitidepsin is making an impact on viral replication in the lungs of this animal. Does it also reduce disease? For that, we look to the lungs of these animals, and we see that in untreated animals, there is a lot of inflammation, both in the alveoli and in the bronchia. But when we treat the animals with plitidepsin, we see very little inflammation. It has been reduced a lot, meaning that there is a big reduction of disease. Remdesivir reduces inflammation, but not as good as plitidepsin. And in uninfected mice, obviously, there is no inflammation. Now, what happens in human? Plitidepsin has been developed by a company called PharmaMar, again for the treatment of myeloma, but they are very interested also in seeing whether plitidepsin is able to be used for the treatment of COVID-19. They have conducted already phase 1-2 trials that shows no severe adverse events and potentially some impact on viral replication in COVID-19 patients. And now they are going to start phase 3 clinical trials. They are already in clinicaltrials.gov being described, which they will involve a total of 600 participants that have been randomized into 3 groups, with one group with 1 dose of plitidepsin, the other group with a different dose of plitidepsin, and a controlled placebo. All of them will receive obviously standard of care. It is remdesivir, and the idea here is to look whether now treatment with plitidepsin results in less days of hospitalization of people with COVID-19. So in summary, with respect to antivirals, I show you how you can use information about virus-host protein interactions in interactome. We have also used the phosphoproteome. I didn't present it here data, but we have some inhibitors also based on proteins that have been phosphorylated. And that provides a rational therapeutic target for drug repurposing.
and how plitidepsin, which is an inhibitor of EYF1A, has potent SARS-CoV-2 antiviral activity both in vitro and in vivo, and phase 3 clinical trials are moving forward. So hopefully they will make also an impact for the treatment of COVID-19 in patients. Now, the second part that I'm going to talk is about vaccines. I'm going to talk about the vaccine that we are developing here at Mount Sinai, which is based on the use of a vector. This is called Newcastle disease virus. Now, in terms of COVID-19 vaccines, there are many different strategies that are being used, and we know that there are already some vaccines that are quite successful, and they're being used all over the world, with some more percentage of vaccinated people in some places than in others, but very successful vaccines. The strategies that have been tried to move forward, they are all the potential vaccine strategies that we know of: inactivated viruses, attenuated viruses, protein subunits, viral vectors like the adenovirus vectors, that there are several vaccines now that have been used around the world, like the Sputnik vaccine, the AstraZeneca vaccine, or the J&J vaccine. Genetic vaccines, among which ones the RNA messenger RNA vaccines are the ones that have been now quite successful and in use in different parts of the world. This is the vaccines from Moderna and from BioNTech, Pfizer. But there are also some other considerations that we need to have with these vaccines. For protein vaccines or inactivated vaccines, the use of adjuvants, the use of nanoparticles, which they have been used for protecting, for example, the mRNA vaccines to be able to be delivered where they need to be, but perhaps also they are useful for protein subunit vaccines. The route of administration, most of these vaccines are being given intramuscularly, but the virus starts infection in the respiratory mucosa, so perhaps if there will be some vaccines that are being delivered mucosally to the respiratory mucosa, they may have a better efficacy preventing not only disease but also infection. And all of them, they are based on the surface antigen of the virus, the spike protein. And it's believed that they are working by using of neutralizing antibodies against the spike protein that prevents the virus from binding to the receptor. But perhaps there is some other cellular immunity that is also contributing to the success of some of these vaccines. Now, the one that I'm going to present is one that we are developing again here in the Icahn School of Medicine at Mount Sinai. My group, together with Peter Palese and Florian Kramer, is basically driven by a senior postdoc in the lab of Peter Palese, Winasan, and it's based on Newcastle disease virus expressing the spike protein of SARS-CoV-2. Now, why this one and why we need the new vaccine? They are quite successful already, the ones that they are. Well, there are some things that we consider that perhaps it's still good to try to develop new vaccines. First of all, there is no pre-existing immunity in humans against this vector, viral vector, Newcastle disease virus, but this is a virus from chicken, it's a virus from avian host. It does not infect and cause disease in mammalian host. It's able to infect cells once, express antigen, and then it's been inhibited very quickly by innate immunity, and so induce a very strong innate immune response. Is very good in order to induction of antibodies because of that. So that's another advantage. But the main advantage is manufacturing. Manufacturing of Newcastle disease virus vaccines. They are being used actually for protecting chicken against severe Newcastle disease virus disease. And they are using the same technology as we use for the production of influenza vaccines, meaning that there is a lot of possibility of production because all the facilities that have been used for production of flu vaccines could be used for production of this vaccine, of COVID-19 vaccine, if it's a successful vaccine. So it can be made very quickly, it can be made in large amounts, it's very cheap to make, and this may make an impact in terms of accelerating the process of vaccination, especially in developing countries. Now, is this a virus, a vaccine vector that is safe? And fortunately, we know of already studies in humans with this virus that has been now being developed as a potential also anti-tumor treatment. And from these studies, we know that it's quite safe the use of Newcastle disease virus in humans.
Now, what we do in order to make it the vaccine vector for COVID-19 is the genome of the virus has several viral proteins, but then we insert one more gene in the genome of the virus, and I'm coding also for the SARS-CoV-2 S protein. This one has been modified to incorporate the cytoplasmic and transmembrane domain of one of the glycoproteins of Newcastle disease virus, and with this we achieve incorporation of the spike into the virus particles, the Newcastle disease virus particles. These viruses are able to express, here as in Western blot, spike at high levels. We use a version of a spike that is highly stable, prefusion, so able to induce the right antibodies that we want to induce in order to neutralize the virus. And this can be produced in eggs, as I mentioned, and now you get viruses that are decorated in the outside with the spike protein, and also when they get inside the cell, will express more spike protein. Now, these viruses can be used inactivated, and that could use the same method of inactivation as the flu vaccine. And they have also the advantage of showing here that perhaps can be also used in live, intranasally. And that may represent also an advantage if it's able to induce much better mucosal immunity. So we have tested the vaccine preclinically in multiple animal models. I'm going to just show you some of the latest results with the hamster model. Hamsters are susceptible of SARS-CoV-2 infection in contrast to mice, and they have morbidity, they develop disease, they replicate the virus to high levels. We have immunized hamsters. In this case, they are immunized with live Newcastle disease virus vaccine intranasally twice, and then challenged with SARS-CoV-2. And what one can see is that in animals that have received controls, then you get the body weight drop due to SARS-CoV-2 infection, but the ones that have received the virus expressing what we call ExaPro spike. ExaPro is a stabilized form of the spike. They are all protected against disease measured by body weight, and they are also protected from a replication of the virus. So if we look to the levels of viral replication at day 2 in lungs, the left, the right lung lobe or the upper right lung lobe, what we see is that there is no detectable virus at day 2, while there is 10 to the 7th PFU in the control mice. At day 5, still no PFU, while there is still around 10 to the 5 PFU in the control mice. And not only protects from infection in the lung, it also protects from infection in the nose, which is what is here down. At day 2 in nasal washes, there is only one hamster that has some level of replication that is lower than the one that is happening in the controls, and at day 5 also there is no measured viral replicative virus in these vaccinated hamsters. So it looks quite promising in animal models. Now we have been now starting phase 1 clinical trials with the collaboration of the governments of Vietnam and Thailand, and we are also expecting to have very soon phase 1 clinical trials started in Brazil, in Mexico, and also here in the United States. We have been lucky to have help from different governments, like the government of Vietnam or the government of Thailand. These countries are very interested in this type of vaccines because they have manufacturing conditions for the influenza virus vaccine, so they can use this for manufacture locally, this Newcastle disease virus vaccine. And they have started, as I mentioned, phase 1 clinical trials in Vietnam in March 15, as well as in Thailand in March 22. Till now, they're going fine. Individuals that have received the vaccine that have not developed severe adverse events, so that looks well. It's a question of looking now at how immunogenic it is with respect to antibody production. If it's as immunogenic as it tells the promise in animal models, you never know, and see whether it can be then used as a new vaccine against COVID-19 that is more easy to produce, and therefore they will be more fast in terms of vaccination in the world. I have a few more slides that just treat the issue of variants, which I think is important because everybody is worried about variants. Now, my lab and many different labs in the world now have tested whether the sera from vaccinated individuals and from infected individuals is still able to neutralize the new variants. And the variants that are of concern, they contain
either of two mutations. One is what is called the Nelly mutation, which is a mutation in the RNA binding domain, N501Y, that makes the virus more transmissible, but also could make it evading neutralizing antibodies, because neutralizing antibodies are against the receptor binding domain. When we test this variant with vaccine sera from vaccinated people or sera convalescent from infected people, we don't see, when we compare the old variant to the new variant, we don't see a drop in titers. That means that this N501Y mutation perhaps makes the virus more transmissible, but still the virus is able to be neutralized by antibodies induced by vaccination or by previous infection. Now, there is another mutation that is present in some of the variants of concern, specifically the ones that the lineages that they started in South Africa and in Brazil, and they are prevalent in many areas of the world. Here in the United States, it's more the UK variant, the one that is more prevalent, which is the previous one, the one that contains the Nelly mutation, still being able to be neutralized by the vaccine. In case of the ERIC mutation, E484K, that is more problematic. The neutralization drops. You can see here the vaccinated against the previous variants. They have very high titers, but they have this with Pfizer vaccine, and then it drops between 4 to 10 times as compared to the previous variants. Now, even though it drops, still the sera is able to neutralize, because the vaccine induces very high titers of neutralizing antibodies. So we think that these vaccines are going to, and actually that's what it seems to be in the clinical observations that have been made more recently. They seem to be also efficacious against the variants, although perhaps a little bit less efficacious than what it is against the previous ones. But a little bit less efficacious is still a lot efficacious, because these vaccines are extremely efficacious for preventing clinical symptoms with COVID-19. So ERIC is a little bit more of a concern, especially for people that have low neutralizing antibodies for whatever reason. They may actually break immunity a little bit better than what it is the other variants. All right, so in summary, I show you how an NDV-based vector expressing SARS-CoV-2 induces potent protection from virus infection, both in lungs and nose of hamsters after intranasal immunization. 2 phase 1 trials started in Vietnam and Thailand, and we expect soon also have trials in Brazil, Mexico, and the United States. And if they show good promise, then we will move to phase 2, phase 3 trials. The new variants are still neutralized by sera from immunized individuals, but some at lower titers, and that underscores the need for continuing epidemiological study, just in case that the virus acquires still more changes that make them breaking better immunity than what they are doing now with these variants. This is just to acknowledge all the different agencies that have contributed in funding research in my lab for COVID-19, NIAID, NCI, Department of Defense, the Bill and Melinda Gates Foundation, some private organizations, non-profit like the Fundación La Caixa, the Open Philanthropy Project, the JPB Foundation. Thanks a lot for all the support for these studies. And obviously, these studies would have not been possible without a group of individuals that are performing work with COVID-19. And here I just want to thank everybody in my group that has contributed one thing or another to COVID-19 studies during this last year. And with that, I conclude my presentation. Thanks a lot